Every hour spent in a loop of digital habits and passive consumption causes a toxic protein to accumulate inside your neurons. This jagged molecular structure physically alters the architecture of your reward system, piercing the cell wall from the within. A stark example of this biological control appeared in 2001 when a group of Parkinson's disease patients began taking a drug called pramipexole. These patients had no prior history of addiction, yet the medication triggered immediate pathological changes. Elderly individuals who had never stepped into a casino began racking up six-figure gambling debts, while others developed sudden compulsions for eating and hypersexuality. Pramipexole acts as a dopamine agonist, binding to receptors in the reward center. In these patients, the drug's chemical presence dictated their choices, overrunning their established personalities with new, destructive behaviors. The medication targeted the mesolimbic dopamine system, a survival circuit reinforcing behaviors like finding food. Modern stimuli possess a configuration that fits these receptors. High sucrose diets and social media feeds generate unnatural spikes in activity along this circuitry. When this ancient system is hijacked, it overrides the prefrontal cortex. It signals to the rest of the brain that the destructive behavior is the most vital requirement for your continued existence. To manage this chemical flood, the brain undergoes a structural adaptation. It modifies its internal chemistry, initiating a physical mutation within the cell. Compulsive habits are the result of a hijacked neural circuit. This biological process structurally alters the foundation of your identity. Dr. Eric Nestler demonstrated that the brain physically transforms in response to both chemical drugs and behavioral compulsions. This transformation is driven by a protein called delta phos-B. It accumulates inside the neurons with every repeated hit of a dopamine spike. Unlike most proteins that degrade within hours, delta phos-B is exceptionally stable. It builds up layer by layer over weeks and months of repeated behavior. Eventually, delta phos-B reaches a threshold that alters the transcription of your genetic code. This epigenetic change mutates the dopamine system, creating a state of hypersensitivity where the brain's physical structure is hardwired to respond to the addictive trigger. This physical state is known as sensitization. Sensitization creates a distinct separation between craving and pleasure. Your brain builds a tolerance to the enjoyment. You like the activity less, but the mutated neurons make you want it more. This manifests as a compulsion to mindlessly open an app or consume a specific food, even while feeling consciously bored or disgusted by the act. Neuroplasticity is the exact biological mechanism that maintains these pathological habits. It is the process that locks you into the behaviors you intend to stop. Neurologist Alvaro Pascual León distinguishes the brain as plastic, not elastic. Once the structure is altered, it never returns to its original configuration. He compares the brain to a snowy hill. Every behavior is a sled run that carves a track into that snow. Compulsive behaviors carve deep, rigid trenches. Biologically, it becomes increasingly difficult to steer your cognitive sled into any other path. In the 1930s, surgeons viewed the brain as a collection of fixed, hardwired locations. They mapped the brain assuming every function was tied to a permanent, genetically predetermined spot. Michael Merzenich challenged this model. He proved that these neural maps are entirely dynamic and governed by constant competition. Merzenich mapped the region of a monkey's brain that processed signals from its hand, then amputated the middle finger to observe how the map responded to the loss of input. The neural territory assigned to the missing finger was immediately claimed by the adjacent maps. The surrounding fingers expanded their representations to occupy the vacant cortical space. This established that brain space is a zero-sum environment. It is allocated strictly by a use-it-or-lose-it principle. This competition for real estate suggests that delta phos-B infected pathways can be starved. If you refuse to use them, those maps can be overwritten by more efficient neural territory. Your brain is a constant war for resources. You have the biological capacity to overwrite pathological loops if you engage the right chemical mechanisms. If delta phos-B creates these rigid biological chains, how do you trigger the neural plasticity required to pave over them? The brain does not reorganize for free. Passive consumption and low-effort habits do not stimulate changes in your cortical maps. To force a rewrite, you must activate the nucleus basalis. The nucleus basalis only activates during periods of intense, unbroken, effortful focus on a novel or difficult task. This activation floods the brain with acetylcholine to sharpen attention and a protein called brain-derived neurotrophic factor, or BDNF. BDNF is a neurotrophic factor that increases synaptic strength. It flags the specific neurons firing during intense concentration and initiates the growth of new dendritic spines to secure those connections. This process temporarily reopens the critical period of plasticity, a state of rapid learning that is usually restricted to infancy. This state requires active, often frustrating effort. It is the direct opposite of the low-effort dopamine spikes provided by digital habits. Curing a brain damaged by passive habits requires you to force the nucleus basalis open through highly focused cognitive strain. Dr. Jeffrey Schwartz applied these principles of rigorous plasticity to treat patients with severe obsessive-compulsive disorder. In OCD, yielding to a compulsion provides temporary relief, but physically strengthens the pathological neural circuit. Schwartz's protocol forces the patient to acknowledge the urge, but immediately redirect their attention to a demanding, positive activity for 30 minutes. This redirection activates the nucleus basalis. The intense focus secretes BDNF, building a new circuit while starving the pathological one of activity. This utilizes the inverse law of plasticity. Neurons that fire apart, wire apart. By refusing to use the delta Phos-B infected pathways, the brain eventually reabsorbs that tissue to support the competing healthy behaviors. Schwartz's protocol serves as a biological constraint. It functions like the physical mitt used in stroke rehabilitation, forcing the brain to forge a path around the damaged circuitry. Those deep tracks in the snowy hill are never deleted, but they are bypassed and overgrown by new, high-speed neural pathways. You cannot simply choose to stop a bad habit. You must actively assassinate it by dedicating your cortical real estate to a new, demanding behavior. Neuroplasticity is an unstoppable biological law running in your head right now. You are currently either secreting BDNF through rigorous focus or allowing delta Phos-B to crystallize your compulsions. Every hour spent scrolling through a feed is a biological choice to further hardwire a hijacked reward system. Maintaining a grasp on these specific mechanisms distinguishes your approach to behavior from the majority of the population. Scrolling past this video without subscribing returns you to the exact passive state that feeds the loop. Every passive click adds another molecular chain to your brain until the person you intend to be is completely overwritten by cut to black.